like there's a patient and they're having something going on,
Yeah.
and I'm like,
Oh, what do I do?
I'm like passed, but I didn't. I don't know what I'm doing, so
No.
I don't even know what that is. Yeah, no, I don't.
Yeah. No. All right. So first of all, since we haven't had like a legit SI session in a while, how's everyone feeling with lecture?
Pretty good.
Yeah.
Good since the last test.
Okay. Cool. Good since the last test. That's a good confidence. Was the last test? Y'all find the last test hard? Yeah. The lymphatic system is like, sometimes it clicks, sometimes it doesn't.
What happens is I go and I'm like confident, and then I go and take the test, and then there's questions that like I just don't understand. I just don't understand them. And then I'm like, oh, okay, I think I get it, and I like pick all the answers, and I'm like, I'm gonna do good on this test, and then I click submit, and then I don't do as good as I want. Every time, literally every time.
It's always the struggle with these kinds of tests. What is the date? It's the 3rd, right? Yeah.
Yeah.
Was there anything in the last 2 lectures, the digestive system, that was confusing? I know you asked me about how hydrochloric acid is made. Did that make sense to you guys?
Yeah.
Oh, Brady, you guys look out here. Okay. Oh, I saw them when I was walking.
Oh,
and she
okay.
was
Yeah.
like, they're up there, and I was like, no. But they were talking. So.
Yeah, I saw them.
Is it
Yeah.
I don't know my student ID number. Is that gonna be like that's fine.
I have.
When you take your teeth,
I'm pretty sure I can see everybody's.
I write mine down wrong like every time.
No, you don't. Well, maybe you do. I don't know. I look up your name.
Some days I don't know it.
One kind of figuring out yourself.
All right. So, so the hydrochloric acid. Well, what's the reaction to make carbonic acid? That's a reaction that you should probably know. Carbonic anhydrase helps make it. It's like an enzyme. But what do you have to? What 2 ingredients? What 2 things have to interact to make?
Hydrogen and hydrogen.
Not hydrogen and carbon.
Oh, to make carbon, right?
Wait,
I'm sorry.
To make carbonic acid. So, because carbonic acid is what releases, thank you, the hydrogen to make hydrochloric acid. So CO2 plus H2O equals carbonic acid. So that's an equation that you should know for this chapter. So I'll write it down because he might put it in he might put it in some form in a description of a question, something like that. So CO2 plus H2O yields H2CO3. And then, so it yields eventually, skipping the middle steps, HCO3 negative plus H positive. Right, so this is our buffer that we've been talking about the whole semester. And then we have this H ion. So when we're talking about stomach acid, why do we care about this hydrogen ion? we going to use it for?
This is what we were talking about.
This is, yeah. I figured we could go over it again because it's getting a little confusing. So, it's the chemistry, it's always a little weird.
Yeah.
You can answer. Hi. Alright, so we're doing how hydrochloric acid is made. We're going over that.
What slide is
Isn't that like, I know that when you take like something like, I don't think it's called Prevacid, no, something. But like,
Parietal or something like that.
right, yeah, something for your stomach that it's like something with hydrogen, so maybe it neutralizes the stomach.
It does. So a proton pump inhibitor
That's what it's called.
or a hydrogen pump inhibitor, because an H plus is a proton. That's all it is. So that decreases stomach acid production. Because when you're making stomach acid, so what is stomach acid? What's the, like, what kind of acid? It's got a name other than stomach acid, like a chemical name.
So, is it corrosive? But that's not.
That's hydrochloric acid. Yeah. So our end goal of the product that we want to make is HCl, right? So look, we have an H. So we've already made, so by CO2 diffusing out of the blood and into these cells that are making the hydrochloric acid, it naturally releases a hydrogen. So we have a hydrogen now, which is great, but how do we get the chloride ion? So I'm gonna draw a little cell. That's what we gotta figure out, is how we get this chloride ion. So here's our little cell. Does anyone remember what kind of cell makes the acid? Parietal cells. Yep, and that is something that you'll want to remember. This is a parietal cell. Alright, so this is a parietal cell. So I'm gonna draw a little blood vessel next to it. So this is a blood vessel. So some of the CO2 is going to go in here, and it's going to react with the water, and we're going to get this HCO3 negative plus the H positive. So this HCO3 negative is a buffer, acts as a buffer in the body. And we mentioned the alkaline flood after you eat, because you're producing a ton of hydrochloric acid. So at the same time, you're also producing a bunch of these negative ions. So we've got this H positive in here, right? But we don't want it in here because we want it in the stomach. Does the stomach have an E on the end of it? No? Okay. I don't know why I decided that.
It's fine.
In my world it does, but that's fine. So we've got to get this hydrogen out, right? So this is where the pumps come in, these proton pumps. So you have little pumps in the wall right here that just transport all these hydrogens out of here, which is great. But we have lost now a positive in here. So now the charge is unbalanced in the cell, right? So what's going to happen is actually a potassium, and this is kind of in here a little bit, but it makes a lot of sense if you look at it in the picture. So there's little potassiums floating around in the stomach. So this potassium will trade places with the hydrogen, and now you have even charges again, right? So, and then there's also, floating around inside the cell, there's also the chloride ions. So both of these, the hydrogen and the chloride, start off in the parietal cell, right? So you've just balanced the charges. So now the stomach, now the little parietal cell says, we gotta get this chlorine out of here. So what it does is actually this potassium that just pumped in, it actually pumps it back out. Okay? So this potassium leaves. So this potassium is literally just floating in and out. It diffuses in and then gets pumped right back out. So now we have too much negative in here. So we could add another positive, or we could take out this chloride. So the cell allows this chloride to follow the potassium, and now the chloride and the hydrogen are both out here, and they just naturally combine and make hydrochloric acid. Does that make any sense? So it's kind of like a game of diffusion that this cell plays. It's like, all right, you know, it's following the laws of nature, right? I've got this hydrogen, I want to get this hydrogen out here, but then I can't be in balance, so I'm going to use this potassium. Then I want to get the chloride out, so the potassium is send it back out, the chloride's going to follow. So the cell is balanced. The cell is fine, and it gets the stuff out that it needs to get out.
Okay. So the potassium is what kind of is making it able to do that?
Exactly. It's kind of a middleman.
Okay.
It's a means to an end. It serves no purpose in here other than, or at least for our sake, other than to facilitate diffusion. So when you're studying that, there's it's a little hard because it's just words on the slide, but if you see a picture of it, it makes a lot more sense, and that's what's happening. So it's a parietal cell pumping out hydrogen through a hydrogen pump or a proton pump. And what is, do you remember what the phenomenon is called when the chlorine moves from one place to another? We saw it in the blood.
Chloride shift.
A chloride shift. So he might ask you something about that too. This right here, when the chlorine balances out charges, it's called a chloride shift. So the three key terms I remember are parietal cell, proton pump, and chloride shift.
Might be ready.
Hello.
Are you recording?
I am recording, yeah. So that's that. It's nothing complicated. Does that make sense?
Yeah.
Okay, cool.
Can you do the next one?
Yeah.
The carcinogen and the pepsin and the, I guess I get live news because like that's what breaks it down.
Yeah.
And then it was like the pepsin and the pepsin, they just like turn
Yes, I just bought one.
Do you have an extra one?
amino acids.
Yeah,
They just
I
break
would
like
like
amino
to
acids.
Yeah, so they break proteins. Proteins are made of amino acids, right?
I have, yeah.
So it's kind of like if you had a beaded necklace and you cut the string.
Okay.
All the beads can now be on their own.
Okay.
Yeah.
Oh, that makes sense now.
Yeah.
What?
So
then,
no, go ahead. Go ahead.
because after that, then it can digest, right?
Exactly. Well, yeah,
Like
they're
in, like-
small enough to
Yeah.
be absorbed and digested. Exactly. Yeah. So it actually brings up an interesting point. So pepsin starts out as pepsinogen, right? And it gets turned into pepsin. It gets broken down kind of like in the blood clotting. Remember how we like cut the caps off of the fibrinogen? It's kind of the same concept with the pepsinogen.
Okay.
But why would the cells release an inactive form of a protein digester?
Oh, he was talking about this. Because you don't need it until, like, because you don't want it to, like, mess with any other imbalances until you need it. Something like that.
Yeah. So if you release an active protein digester, what are cells primarily made of? Protein.
Right.
So if the cell makes pepsin
Mm-hmm.
in itself, the pepsin will start digesting the cell that's making it. And all the cells in the channel leading up.
Okay.
So you want to release an inactive form, and that's the same concept with all these chemicals that you'll see. They get released like when you're studying it, if there's an inactive form, it means that you want to release it inactive so it doesn't digest itself before it gets to the substrate.
The chloride in the stomach,
The hydrochloric acid.
yeah, the hydrochloric acid in the stomach that activates it.
Yes, so it breaks it. It does the same thing. I know, because remember fibrinogen,
Right.
the gin part
Yeah.
is like the cap. So it's kind of the same concept here. You kind of cut a piece off, and now all of a sudden the active spot is released, and it can go ahead and just eat everything.
This makes so much more sense.
Yeah,
Just talking about it.
yeah, it really does. So it has to be broken down before it can break anything else down, which is interesting. So I figured for people who just walked in, I figured we'd go over lecture first since we haven't really touched much on lecture, and then once we get all our questions out of the way, then we can do lab stuff if y'all wanted to. Y'all look very stressed, all of you guys.
No, we're not nervous at all.
Like you guys are all just like staring at your papers. Did y'all have any questions about the digestive system? It's pretty straightforward. It's mostly just memorizing anatomy and where each chemical is released. And if you can do that, that's pretty much it. Yeah, there's not really.
Yeah, I'm looking through it now. There's not too much.
No, it's mostly just memorizing. Malt. Yeah. So what does malt stand for? Mucosa-associated lymphatic tissue. So what does that mean? Where is this lymphatic tissue located?
In the GI.
It's located in mucous membranes. So malt tissues are found in the mucous membranes. So you actually have malt tissues lining the opening to your throat so that the white blood cells can look at everything that you're swallowing, that you're breathing in, right? So what's the other name for the malt tissue in your large intestine? The Peyer's patches.
Okay.
Yeah. So you have Peyer's patches, right? Because what are you doing in your GI tract? What's the primary thing you're doing? Absorbing. So you're absorbing all this stuff. So it makes sense that you want extra special protection. You want screening. It's a border crossing. You want border police, border patrol to make sure that everything coming in is legit and okay. So that's why you have the Peyer's patches, to make sure that that when you're absorbing all this stuff, that you're not absorbing things that are going to kill you.
And if it's bad, then you just don't absorb it and it just continues growing?
It would, yeah. I mean, I think it would depend on what it is, like as to how the body would react to it.
Because then you have stereotypical reactions. Okay.
Yeah. If it's something, I'd imagine if it's something the body can selectively not absorb, it would shut down those pumps. But if it's not something that it can selectively absorb, it would just kill it, like right away. Yeah, but that's what you have the malt tissue.
So
it's just the Peyer's patches.
It just means the mucus tissue. That's all.
Yeah, it's lymphatic tissue. So it's lymph nodes without the capsule, just in mucous membranes. Mucosa associated lymphatic tissue. That's what it is. Also, the amount of fluid produced by the digestive system is just disgusting.
Crazy.
Like, I'm not gonna lie.
border enzymes.
Mm-hmm.
Those are just like, they just like what you just said, like they like monitor and
They-
keep things in, they call them, so they're regulated.
So they're just on the surface of some cells, so that when food brushes up next to those specific cells, it acts on that little bit of food. These are not enzymes that you want to, you don't want to spit these enzymes into the whole lumen, because there's some parts of the lumen that are unprotected from these enzymes, so you digest yourself. So you keep it on the surface of this protective cell, the cell that's protected from it.
Sorry for the question.
No, you're good. That's why we're here, is questions. Yeah, because these are not enzymes you want to spit into the entire intestine. You want to keep them localized, but you still want them to work. And then the large intestine is like really boring. I actually forgot that Hostra and Tinea coli were things until like right before my practical, like literally 10 minutes before. Someone mentioned it, I was like, wait, what? And they're like, the Hostra, the Tinea coli. I was like, I don't remember that.
The hostra is like the digits, right?
It's like the bulges in the large intestine.
Oh, yeah.
And then Tinea coli is the smooth muscle. The band. It's like,
Oh,
yeah,
wait, let me see.
that right there. The little band.
Okay.
Anything else about the GI tract? So who has a practical today? Oh, there's a lot of people, okay. All right, so is there anything, and if you think of anything about the digestive system, anyone, you can pipe up, whatever. So anything specific that y'all are worried about the practical?
Everything.
Everything. Well, I can't go over a whole semester in 30 minutes.
In the beginning.
Right.
Sophia created the class. And it was good.
Is there anything, so narrow it down a little bit. Like anything that you, endocrine, okay. So,
I'm trying to take over.
alright, so I have one vote for endocrine. Anyone else? So the endocrine,
We can talk about
okay, so the endocrine, really for lab, the endocrine is literally like, where is this gland and what hormone does it release? You already did all the physiology in the lecture, so you don't really have to worry about that, the function of each hormone, like you did that. So it's mostly just learning, this says, identify the hormones which are secreted by each endocrine gland, be able to, you'd know the general function of the hormones already. So, what's a tropic hormone, first of all?
Aren't those hormones that, sorry,
No, you're good.
aren't those hormones that like cause something else to be released or something?
They cause another gland to release another hormone. Yep. And then there was that chart that he gave where you could list all the hormones and their functions and their location. That's like your holy grail of hormone studying. That chart right there.
I probably carried this around with me everywhere I went for like 2 weeks.
Let's see. So I'll just throw out some random questions. Where do you find your adrenal glands?
On top
on
of
top of the kidney.
On top of the kidneys. What does the adrenal medulla release? And norepinephrine, exactly. Yep. So that's part of your fight or flight response, right? Yep. So then what does the cortex release?
Androgens? No, those are. What are we talking about? Adrenal?
They're the adrenal cortex. So those are your corticoids. Your mineral corticoids, your glucocorticoids, your gonadocorticoids, which are your androgens. Yeah. Yeah, so androgens. But if you see, so your cortex is corticoids. If you're asked about the cortex, and one of the, there's an option that's corticoid, anything corticoid. It's your medulla, or your cortex. Oh my gosh.
But then he's not gonna put like corticoid on there. He's gonna put the other name like aldosterone or cortisol or androgens.
He could, or he could say what gland releases your mineral corticoids.
I mean, just because that's more difficult for us to remember.
The mineral corticoids?
No, the other names.
Oh, the other one?
Versus...
Yeah. Yeah. It's either or. You could do both. What does your pancreas release?
Insulin.
And glucagon. What does your thymus release?
That one's not really deep for that. I wrote that one.
I think you're thinking of thyroid.
I was thinking of thyroid.
Isn't that T cells?
Peptide hormones.
Thymosin and thymopoietin. I think those are peptide hormones, yeah. But thymosin and thymopoietin are the answers. So remember, the thymus is where the white blood cells mature. So the thymus is in charge of making sure that they're made and that they mature. That's what these hormones are for. So what does the thyroid gland release? T3, T4, and calcitonin, exactly. And parathyroid.
I'm sorry, what did you say the thymus is for?
The thymus is for thymosin and thymopoietin. So white blood cell production and maturation.
Okay, thank you.
Yeah, parathyroid is literally parathyroid hormone. It's the opposite of calcitonin. And then, what hormones does the posterior pituitary synthesize?
Oxytocin.
Nothing.
No. Oh, it's in the anterior.
Yes. The posterior pituitary synthesizes nothing. It's just a storage area. Yep, so ADH and oxytocin come from the hypothalamus, but they're stored in the posterior pituitary. So be on the lookout. That's most definitely a question he could ask. Is what does the posterior pituitary synthesize, or what hormones does it make? And the answer is nothing. But if he asks what it releases, the answer is ADH and oxytocin. So it's an easy one to get if you remember. It's an easy one to miss if you don't read the whole question. It can be, but it's a good concept question to test whether or not you understand what's going on, which is why be on the lookout for it. And then what are some anterior pituitary hormones?
Is that ACT?
Hormone.
No.
ACTH?
TSH, FSH,
Yeah. And then your hypothalamus has got the tropic hormones. Yeah. So it's easier to remember. Honestly, it's easier to remember what the either remember what the anterior pituitary releases and then remember everything else is not anterior pituitary. And a lot of the other hormones just sound like where they're released. Like if you look at something like thymosin, If it's like, where's, what produces thymosin, and you're like, oh my gosh, this sounds a lot like thymus, is this a trick question? No, it's not. It's the thymus.
Yeah.
Yeah, I mean, that's pretty much what the endocrine is identifying where and what.
And you want to do it next time?
We could do blood next.
I thought we were covering blood because we didn't take a look at it.
There will be, there will still be blood stuff, like
He's gonna have
blood
this-
typing.
He said he's gonna have cells on there.
Is he gonna have,
Like a neutrophil?
Yeah.
okay. He's not gonna have them in, he's not gonna have them in a microscope on slides, because people move the slides, and then you can't see them.
Then you mix.
The blood handout.
So we're doing the white blood cells and stuff like that. What it means.
Well, anything, yeah, so he could ask you about, what white blood cell does what function, he could ask you about red blood cells, so, like, what's the lifespan of a red blood cell?
Is it like 120?
120, that's the average. Yep. Let's see. Well, what are your A granulocytes?
Isn't that like neutrophil, right?
That's leukocyte and
Monocytes.
monocytes and lymphocytes.
Lymphocytes and monocytes. So what is a leukocyte?
White blood cell. So white blood cell.
White blood cell. Leukocyte is the general name. Leuko means white.
Mm.
So leukocyte is a white blood cell. A lymphocyte, I get them confused and mixed up in my head too.
Yeah.
Lymphocyte is an A granulocyte. And then, what are your granulocytes? Yeah. So what's the order of most common in the blood?
Never let monkey eat banana.
Never let monkeys eat bananas. Exactly. Yeah. So neutrophil,
It's not leukocyte.
lymphocyte, monocyte,
eosinophils,
basophils. Exactly. Yep. And then if you're, let's see, I'll let if you want to later look at the blood handout, he has a couple of notes on how to identify them. If you're looking, if he shows you pictures, which I'm not 100% sure if he is.
That's what it said.
Okay. Okay.
He
Yeah.
said it could be on there.
Okay, yeah. So if you see a cell that stains red, a white blood cell that stains red, always the eosinophil. Eosinophils are the only ones that stain red. Everything else is purple. Yeah, it's kind of a little trick. It's like dark red, so it's not like bright, vibrant red, but it's definitely reddish. What cells do platelets come from?
Megakaryocyte.
Megakaryocytes. Where are the megakaryocytes located? In the bone marrow. So where are all these cells made? Bone marrow. Yep. All of these formed elements are made in the bone marrow. Let's see. And then he could ask you about blood typing too. So, so what's the universal donor? O negative. O negative is important term. It's O negative.
What is this RH on it?
Then it's not the universal donor.
Oh.
So O positive means that it has the positive. It has the RH on it. And if you donate O positive to a blood type that doesn't have the RH, that blood type will see the RH and will attack it anyways.
Okay.
Yep. So O negative means that there's literally nothing on that cell that will react with anything with any other blood type. And then what's the universal recipient? AB positive. Yep. AB positive. He will RH is going to be a factor in there. Like he'll put RH and stuff on there, and you'll have to you'll definitely want to make sure that you're at least a little bit comfortable navigating around the RH factor too. And he could ask you, like if someone has A negative blood, who can they receive blood from? What blood types?
A
A
negative.
negative and O
negative.
O negative. Yep. Yeah. So things like that. Not like there's not a thousand things in there. That's kind of an overview. Let's see. And then if he shows you pictures, you know, monocytes are huge, right? They're gigantic and they have like an M-shaped nucleus, kind of an M or like a really big bilobed nucleus. Oh gosh, I got off the internet. The internet sucks. Anything else? I mean, what next I should say?
I wrote diapedesis underneath the granulocytes on my note page. I'm just wondering how that might be related to. Not to be confusing.
No, you're fine. I'm gonna look up diapedesis. Okay. Yes, diapedesis is when white blood cells that are floating along in the blood go through the capillary walls into the tissues. So ped in there means feet. That's when they use their little feet to like grab onto the wall and like pry the wall open and get out.
Okay.
Yeah. So it's like it has something to do with feet, but I didn't remember off the top of my head.
So anything for you.
Thank you, Rainy.
Let's see. What else? First of all, I think y'all are gonna do fine. I'm just putting that out there. I think y'all are gonna do great.
When do we get our results?
Next week. He's not gonna release it. He's not gonna release results before everyone's taken it. So I would expect them.
Maybe Friday if we're lucky.
Yeah, Friday possibly. Monday is also a possibility. He does let people go to his office, and if you go up to him, you say, "Can I look at my practical?" You know, he might let you. But he just doesn't want to release all the grades so that people can't cheat. It's like if people get their grades from the practical at 2 today after the practical, then anyone who remembers questions can be like, "Ooh, I got a 95, which means I was mostly right. So let me go talk to my friend who's taking it on Thursday and tell them all the answers I remember." Yeah.
For one of the models, it was this one.
Mm-hmm.
And is that this one?
Yes. Yeah. It's the digestive one. So
What am I looking at? I was trying to study and I was like, I'm like, it's fine.
You're looking at a very, very small section of the small intestine wall with the villi.
Okay.
So you're looking at the blood vessels and the lymphatic vessels that the nutrients is being absorbed into.
Okay.
So what's the green vessel?
Is that lymphatic?
Lymphatic. So lymphatic, generally, like in that model, especially if it's green, it's lymphatic.
Okay.
So that's kind of just like that. The green splotch in there is the Peyer's patch. Lymphatic. The green vessel is a lymphatic capillary. Generally, most people, most artists stay with lymphatic is green.
Okay.
So if
There's
you're
that one that's purple.
Really?
It's one of the models. It's like everyone is green and then that one's purple. And
Oh, it's like
is that the hepatic portal system?
Yeah, it's the one. Is it?
Because that's not technically lymphatic,
Oh. Maybe
but
it's
yeah, in the big person models, the hepatic portal system is purple
Yeah.
versus yellow, like it is in the cat.
Do you know like? Yeah.
Let's see. Oh, how can you tell if a heart, if you're looking at a sheep heart, right side up?
the pulmonary, would it be like the pulmonary? No, that's anterior.
You can't always see on these sheet parts, some of the vessels are cut super close to the top. So what is your key to look for?
Would it be like
The muscle?
Not the muscle. So you want the apex to be on the bottom
Yeah.
and the base to be on the top, because there's no guarantee that he'll give it to you right side up. He can give it to you upside down.
Okay.
So when you look at it, don't just start answering questions. Give yourself like 5 seconds. And I know, there's not a lot of time, but it makes it worth it so you don't get it backwards. So look for the base has to be on top and the apex on the bottom. And then make sure you can identify left and right. So how do you tell if it's left?
Just turn around.
Yeah, that's what I'm saying. That's what I was saying. The muscle, like it would be thicker.
The muscle is thicker, the ventricle is bigger, and then the right is smaller, and the muscle is thinner. Exactly. Yeah, so look for up, down, left, and right before you answer a question.
A-B. Sorry.
Huh?
Sorry. I was gonna go. You did it up, down, left, right. I said A-B, A-B, start select.
Yeah, that'll just help you make sure that you don't, start answering questions and then halfway through you realize, oh my gosh, this thing's upside down. Or this thing is left, backwards, left and right. I've been answering left to the right.
That's a little hard.
It's a little pro tip.
What? So is this
Um,
I know.
let's see. Anything else?
One of the elements is just white or red blood cells, right?
And platelets. Yep, formed elements, white, red, and platelets. The formed elements that form in your red bone marrow. And the non-formed elements are like the proteins that float around in your blood, the plasma, the enzymes, the CO2, stuff like that.
So what you do is, you know how you have like there's a couple minutes to study before you have like
Because it's like,
if you study like those couple minutes.
And then the ECG wave, just know like P, Q, R, S, T,
Yeah.
right?
Wait, what?
No.
The ECG wave.
PQRS.
Oh, yeah.
Yeah, isn't it? It's
So what is that measuring first of all?
the heartbeat or the cardiac cycle.
The cardiac cycle, but what, how is it measuring it? What is it tracking?
The electric.
Yes, it's tracking electrical differences, repolarization, depolarization. That's why it's called electrocardiogram. So, oh shoot, what did I do? Okay, never mind. So what is the P wave? Atrial depolarization is what'll probably be the closest to the actual answer. Atrial depolarization. What's the QRS complex? Ventricular depolarization. And then what's the T wave?
Ventricular repolarization.
Ventricular repolarization. So what does depolarization mean?
Contraction.
Contraction. What is the other word for contraction? Systole. Yeah. So we saw atrial depolarization and then ventricular depolarization. Where is atrial repolarization?
The ST.
It's hidden in the QRS complex. It's too small. Because the ventricle is so big, there's so much electricity going through it, it just overwhelms the ECG and takes over. So the atrial repolarization is hidden in that QRS complex.
So then what did the PR do again? I know PR interval, what is
The PR interval. It's the time between atrial and
the ventricle?
It's just like the two together, right? P wave and PR segment, flexibilization of what generate atrium.
yeah,
It's just like
because it's a little break because you want the atrium, you don't want the ventricles to depolarize and contract immediately right as the atrium atria are. You want the atria to squeeze the blood in first. You want to give them a hot second, and then the ventricles can take over.
Is that why there's also like a pause for the AV node?
Yeah, that's why the AV node, that's why it goes really fast, right? For the SA node, it spreads really fast to both atria, and then it lets them contract, and then the AV node is slow to allow the blood to drain out. And then once it gets through, what's the next part? After the AV node? The bundle of His. Once it gets the bundle of His, it goes really fast to the Purkinje fibers. Yep. Exactly. See, now we're gonna do fine. Now we're gonna do great. Let's see. And I mean, that's probably only going to be a couple of questions, but it's a couple of, if you understand what's going on, not too difficult questions. And then generally anatomy, like knowing where everything is.
I mean, that's what most of the questions are.
Yeah, and especially I think in anatomy too, because, we did a lot of like cellular, and we did a lot of histology in A&P I with all the tissues and stuff, but in A&P II it's more big picture, like organ systems. So knowing where and what each organ is important, and in my opinion, it's a little more anatomy, I think.
Could you go over, could you go over like the blood vessels? Like
The blood vessels?
Yeah.
Let me pull that up. Like the anatomy of them?
Yeah.
Okay.
Like that.
Okay. So let me orient myself real quick. All right. So a trick that my SI leader taught me is when you're looking at the 3 arteries coming off the top of the heart, you can do this with your hand and put it right here, right? So this is your hold on, I'm orienting myself. Gotta make sure that, because I'm looking at it from a different way. This is,
The middle one's your common.
I'm blanking. Yes, thank you.
You're welcome.
Brachiocephalic, because this is my left.
Wait.
Yeah, coming off of the aorta. Yeah, brachiocephalic, and then this is your carotid, and then this is your subclavian.
Yeah. It's so confusing because it's backwards. I don't think that helps me.
No, I think I did it backwards. It helped me on the test. If it helps you, great. If it doesn't,
I just know. It's just
okay.
really going
If
over.
you can orient yourself with it, then it helps. I think I'm gonna confuse you if I do it, because my left and right is not working today. Anyways, what's the artery that goes under your arm? Axillary. Because this is your axillary region, right?
Yeah.
Mm-hmm. Yep. And then what's this artery? Radial. Mhm. And this is ulnar.
So they kind of align with the.
They do. The arteries and the veins generally, yeah, your brachial is
The here one that's up here.
So,
It's brachial.
well, your brachiocephalic are your arteries. They're like right up here.
Yeah.
Okay, just
Yeah.
like PU.
So generally, the arteries and the veins, like if you're looking at it and you're blanking out, it usually follows the bone that it's on. So, like, what would this artery be? Femoral, and this one? Popliteal, because the back of your knee is your popliteal region, right? And then, what is this one?
Tibial.
Yeah, your anterior tibial.
Yeah.
So, So the ones in like the your arms and legs follow the bones that they're on.
And it's the artery.
And veins. The veins do that too. And then what are the ones on the insides of the arteries on the insides of the one? So the aorta branches off into
The renal.
like right to go to your legs. That's what I meant. The iliacs, yeah, the common iliacs. And then what do those branch into?
External and internal.
External, internal. Which one turns into the femoral?
The external?
External. Yep. Exactly.
So this yellow thing in the cat was this straight
The yellow?
So
It's lymphatic.
It's a lymphatic one.
It'll be lymphatic.
Is the yellow one lymphatic? The yellow?
the yellow
It's a
is
The big yellow on the cat.
the yellow is the it's the hepatic portal system, I think.
Oh.
I don't think you can see the lymphatic in the cat.
Okay.
I think it's all
not
too small.
pricing.
I think it's too small for them to stain it.
Okay.
Yeah, so in the cat, that doesn't sound wrong to anyone, right? The lymphatic is, or the hepatic portal system is yellow.
Yeah.
Yeah, the hepatic portal system's yellow. So if you're asked about, a yellow vessel, just point to a yellow vessel, eliminate everything that doesn't have to do with the hepatic portal system, and then go from there.
They're all gonna be just different parts.
Huh?
He's gonna do that to us. They're all just gonna be different parts of the hepatic portal system. You have to choose one.
Well, I mean, it is the nice thing about the cat is that things are color coordinated. Like you can tell what an artery is and what a vein is because it's color coordinated, which is really nice of them because you could just throw a cat that's brown on the inside
Yeah.
and just like gross looking.
But in the lungs, isn't it flipped?
Yeah, the colors
Like the
change.
colors are supposed to change in the lungs.
Yes.
Like the
The
arteries
vessels-
are blue and the
Because
The
what
veins are-
what is an artery? What's the how do you know it's an artery? Away from the heart. So what's a vein?
towards the heart.
Towards the heart. So do arteries always carry oxygenated blood?
No.
No, exactly. That's also a question you could throw out. Arteries always carry oxygenated blood, true or false? Answer would be false.
False, yeah.
That's actually one I got wrong on one of my quizzes. I was so mad at myself. I was like, oh, I know this. I was like, why did I do that? And then my professor graded our quizzes right in front of us. And I was like, hold on for a minute.
Pulmonary artery goes to the lungs. So it's deoxygenated.
Yes.
Okay.
Yep.
Sorry.
Yeah, so when you're looking at the heart model or the lungs or anything, don't rely on red and blue. Know the path of it's like if you rely on red or blue, you are likely to get at least half the questions wrong, or get half of them wrong, because half of the blood vessels, half of the arteries are blue and half of the arteries are red. So so it's easier. It's just you'll get more right if you know the flow of the blood. So
No pressure.
Well, what side does deoxygenated blood from the body come into the heart? The right side.
Yeah.
Yep. And then it goes to the pulmonary trunk to the lungs. Into the left side. Through the
pulmonary vein.
the pulmonary veins, which are the four veins on the back.
Right, and then out the
Out the aorta. Exactly. Yep. Exactly.
got it.
So if you know that flow, you know what's a vein and what's an artery. Like even knowing that general flow.
I don't think you're.
I said no,
You
no,
said
no,
no,
no.
today.
Yeah. So
Otherwise, I'm sure Victoria will come down with that.
What begins?
So
Yeah, the
I'm taking tomorrow off
on the right
homework.
side.
from where's the oxygenated blood coming from?
For my school tomorrow.
From the body, yeah. So the body brings up through the vena cava to what side?
Right side.
The right side. What chamber?
You plan
When you
it.
take it, because next
The
week's last
atrium.
week.
We're gonna be taking
Oh.
it next week.
Mhm. What is the aorta?
Brittany's gonna take hers, and I'm gonna.
Is that not the
Are you
actual
a
part
doctor?
itself? Like,
Oh,
That's
okay.
the major
Oh, the-
blood vessel. Yeah, the
Yeah.
big blood vessel that arches and goes down. That's the aorta. So
to the right atrium,
right atrium.
When I was
What
going
valve?
to do the LPN program, and then I switched out to the RN.
Is there a tricuspid?
The tricuspid valve. Yep.
I know it's
To
only
the
twice per year, but like, it's like that.
Right ventricle.
Next year,
Through what valve?
Yeah.
So that's why I'm not.
Which semilunar?
Yeah.
Times.
No,
You got that much money?
No, he just said.
no.
Pulmonary semilunar. Because it's going to the lungs.
lungs.
So then what's the blood vessel that it moves out of?
But you only got twice a year.
So I figured
And you
I'd.
gotta wait 30 days.
And then I,
You don't can do
you
it.
know, come back next week.
Pulmonary trunk.
okay,
Yeah. Yep, pulmonary trunk. And that's a blue artery. So remember that. And then it comes, so it goes to the lungs, and then it comes back through
A 58 point, like, 9, because I was so close to the 59 that you gave us.
the four veins.
the four pulmonary veins,
Yeah, the four pulmonary veins into which chamber?
It was the whole thing.
left atrium,
The left atrium through what valve?
Yeah, I thought it was
the
62.
bicuspid
The bicuspid valve to
I haven't seen.
left ventricle, and then
but
out
when I was taking it,
to the rest of the
I
body.
just wanna know
what
it is,
vessel does it go
what
through?
I'm expecting.
you get a 69.
Is that why
63. Don't you need a 63 for reading now? Or is it a 60? I
I think
don't
it's
know.
63 for those two, but yeah.
So it goes through the aortic semilunar to the aorta. So I always remember there's two semilunar valves, right? And two cuspid valves. So the AV valves, so the tricuspid valve is
Yes.
another name for the right AV valve, right atrioventricular valve. So the AV valves are on the bottom, and then the semilunar valves are on the top. I was remember the hood is above me. The hood is above me, so the semilunar valves are on the top of the heart. And then the semilunar valves are just named for what vessel they open into. So pulmonary semilunar goes to the pulmonary trunk. Aortic semilunar goes to the aorta. Yeah. Yeah. So the heart is named, the valves and stuff is named for where it is. So it's kind of duplicated on both sides. It's got the same structures on the left and the right. You just have to remember which one's left, what's the name for the left, this one's on the right, what's the name for the right. That's it. Yeah. Stay good. Alrighty, it is 12:04. Anyone who wants to leave is free. I can stay if y'all want me to.
But yeah.
Pause the video. Huh?